Welcome back to this course on organic chemistry in biology and drug design. Last time we had a very basic understanding of the genesis of this course, and then we started with what are the molecules that are essential for a living organism to have. Now, these bare essential molecules of life are actually the proteins, the nucleic acids, carbohydrates and lipids. Now, these molecules are also called molecules of life, rightly said, because without these molecules the living system cannot survive. Okay, now, because most of these molecules are having molecular weight very large, unlike the small organic molecules. So, sometimes they are also called biological macromolecules, like proteins. They have a building block which combine with each other and finally form the proteins. Same is true for nucleic acids. For carbohydrates again we have building blocks which are called monosaccharides, These are the building blocks. For lipids there are different types. There could be building blocks to make the lipids, or there could be a single molecule which is biosynthesized from very small starting materials, like a derivative of acetic acid which is called acetyl coenzyme E. Lipids is slightly different from the other three categories. The other three categories are all macromolecules. This is a macromolecule, where the building blocks we know that they are amino acids. Nucleic acids, they are also macromolecules and their building blocks are nucleotides or deoxynucleotides. We will come back to these nucleic acids elaborately when we describe the DNA and the RNA molecules. Carbohydrates are also made up of, monosaccharides, but interestingly some of these carbohydrates are also present in the form of monosaccharides in the body. Like we measure the glucose level, we know that is a test that what is a person is suffering from diabetes or not. So, we measure the building blocks which are also present and having a biological function. On the other hand, these building blocks are joined together and stored like what is starch in plants. So, these are also stored, and this storage material is called glycogen in case of humans. This glycogen is nothing but again combination of glucose through various glycosidic linkages. Only thing, it is more branched than the starch. So, at the time of when glucose is in short supply, then we can break down this glycogen and get the monosaccharides, which can be burned later on to generate energy. Lipids on the other hand are really, it is a varied class of molecules. Now, the definition of lipids is basically the molecules which are not water soluble. They are called lipids. They are actually soluble in nonpolar solvents like the organic solvents. Okay. Now, there could be several molecules like that. Some of the lipids are actually derivatives of fatty acids, and some of the lipids could be steroids, and there are many other molecules which are also like terpenes in plants. They are also soluble in organic solvents. They are all mostly made up of hydrophobic hydrophobic carbon chains. Okay. Lot of carbons are usually there and functionalized carbons are much less in lipids. I will show some structure of these molecules of life, but before that let me again point out something, that last time we said that in addition to these large molecules, what we have also, there are some small molecules which are also essential for the survival of the living organisms. Like some molecules are say coenzymes, which are small molecules which assist the enzyme to perform their job, that means to catalyze the reactions. There are neurotransmitters which performs the signal transduction. Okay. There are hormones. So, these are small molecules, some of them could be lipids, because they are insoluble, by definition that they should be lipids, because lipid is which is insoluble in water. So, some of them are lipids, some of them could be amino acids also, some of the neurotransmitters are amino acids like gamma aminobutyric acid. So, they are a different class, that is the small molecule domain of biology, and there is this large molecule domain of biology which consists of proteins, nucleic acids, carbohydrates, and lipids. In this case, lipids which we are talking about, fairly large molecules consisting of big fatty acid chains. Okay. Now, let me show you some of these molecules of life, like proteins. Now, proteins again, building blocks, we know that amino acids combine. When they combine, they expel a molecule of water and form a bond which is known as the amide bond, in this case called a peptide bond, because what is you are making is a peptide. Okay. And the ultimately how many amino acids will join, that depends on the nature of the protein, and the direction has to come from the nucleic acids, because there is a hierarchical structure in biology, that although we have these proteins, nucleic acids that they are there, but we have a hierarchical structure, means that who gives the direction to make a particular protein, and that is, it is the nucleic acids. So, from nucleic acids the information flows up to proteins. This is also called the central dogma of biology. We will also again come back when we discuss nucleic acids, because nucleic acids are also of two types, the ribonucleic acids, the RNA, and the deoxyribonucleic acid, that is the DNA. Okay. The central dogma says that it is the deoxyribonucleic acid polymers, they are the ones which sends the signal via the RNA, which is called the messenger RNA. They take the message and then translate the message into the proteins. Okay. And however, there are certain violations of this central dogma. Some viruses follow, they only have RNA, the ribonucleic acids, they do not have the deoxyribonucleic acid. So, in that case the central dogma is not applicable, but anyway those are very special cases. Now, let us show these molecules of life, how do they look like, what is their three-dimensional structure. This is a beautiful structure of a protein which has been crystallized, and its crystal structure is what is shown here. Okay. So, you see there are lot of folding that has taken place, because in this system if you take the two terminals and then stretch them. So, the length of the protein will be very long, and it will be very difficult to have this protein ultimately fit into the small cell that we have. Okay. Because ultimately if you break down the biological system, so the hierarchical is that, suppose the first it is the organs, then tissues, and then from tissues it goes to the cells. So, after the cells, then the internal structure of the cell. So, cell has a definite dimension. So, the proteins cannot be just have a linear stretched chain, that is number one. However, that is not the only reason why proteins will fold. It folds also in such a way that it generates certain pockets, which pockets are important to do whatever the function they are supposed to carry out. Okay. So, this is the crystal structure of a particular protein. We see that we know that this is composed of some, I could see some helix, you see that the green one and the red ones, and then it has got certain portions which are called sheet structure and certain are random loop structure, okay, some like this loop what we are seeing here. Now, that is the protein. Now, next let us show some carbohydrate. Now, carbohydrates, as I said, is pretty interesting because we also use the monomers, that means the monosaccharides, as well as we store that into the glycogen at the time of need. Okay. It all starts with, see, this is the glucose molecule. Glucose molecule is shown here. This is the simple monosaccharide that we have, and glucose we know that it can be present in the alpha form and the beta form. There are two forms of glucose which is shown here, and we also know that there is a if you take glucose and subject it to a pH, an amphoteric pH, then what happens? There will be what is called a mutarotation. Amphoteric pH means I am saying that there is acid and base both are present. Then in that system, the glucose, if you take alpha glucose, that isomerizes to the beta glucose and vice versa, and finally an equilibrium that takes place, and this process is called mutarotation. However, the point here it is that just to show some of the carbohydrates that we require in the biological system. Now, remember this is the monomer of the carbohydrate, the polymeric carbohydrate that we are talking about. Now, sugars can be, again, usually sugars are polyhydroxy, aldehydes or ketones, but sometimes in these biological systems what we find that some of the carbon lacks the hydroxy unit, then we call that as deoxy sugar, and this is a very vital step that is happening in biology, because we know that ribonucleic acid has a sugar unit which is ribose, and this ribose in RNA has all the hydroxy groups what they are supposed to have, and in the deoxyribonucleic acid you lack the 2' hydroxy. Okay. If you know the numbering, you must be knowing, we can again go back that the 2' is next to the anomeric carbon, and that is missing in the DNA. So, this has to be, because DNA is the fundamental molecule in our hierarchical system, hierarchical biological system. So, it must be a very important step. Okay. And so, we have shown this glucose, and glucose can also be see it can present there are 5 member rings also. Apart from glucose, we have ribose. Ribose is the C5 5 carbon containing molecule, and the structure of ribose is shown here. It has got a furanose structure. By the way, if you have a 6 membered ring, that is called a pyranose structure, and if you have a 6 membered ring, that is called a furanose structure. Okay. So, ribose is present usually in the furanose structure, and here on the right hand side it is shown in a furanose structure, and it is also a deoxy. It is precisely, this is called alpha D 2-deoxy ribofuranose, and the arrow points out where is the deoxygenation that has taken place, and why it is alpha, because the anomeric weight which is adjacent to the ring oxygen, that is the anomeric carbon, and the weight is alpha, that means below the plane of the ring, the way it has been presented. Okay. There are other forms of carbohydrates where the hydroxy group may be replaced by an NH2 group. Like in this molecule, what we are seeing is that it is a glucose molecule close to glucose molecule, but only one difference, and that is the 2 prime, the 2 prime carbon. Of course, in glucose there is no prime. We are talking about this prime number in relation to a nucleic acid, because in nucleic acid you have a base, you have a sugar, and you have the phosphate, and the base takes the numbering 1, 2, 3, 4. Then the sugar gets the numbering 1 prime, 2 prime, 3 prime. But here it is only glucose. So, we cannot really, technically we cannot say that this is a 2 prime position. We have to say 1, 2, 3, 4, because this is only the carbohydrate portion and not the nucleic acid. So, this is that 2 amino, basically 2 amino glucose you can say, but it has got a name, it is called glucosamine. And this is beta, you can see it is beta because the anomeric OH is in the beta orientation above the plane of the average plane of the ring. Okay. And also in the beta form, this is in the equatorial position, and the NH2 is in at the 2 position, okay, that is alpha. This is a very important molecule. We will see later on the function of this glucosamine. This is a part of the unit which forms the cell wall, the wall of the bacteria. The cell wall which contains the bacterial material inside, that cell wall, the glucosamine is a constituent of that, and which is a little bit shown here, that the glucosamine ultimately goes to the acetyl form, the nitrogen is now acetylated as you see, and then there is some serine, then there is a protein chain. So, this participates in the formation of the cell wall biosynthesis, but we will come back to that when we talk about the antibiotics and their action on bacterial cells. This is what I was talking about, this glycogen, this dotted structure, that dotted structure on the right, that is what is shown as here. This dotted structure, the branch structures, that is what is glycogen. Each dot is representing a glucose unit. Okay. As I said, this is just a counterpart of starch which is present in the plants. Okay. The carbohydrates, so what is the function of carbohydrates? There are many functions. One is the carbohydrates are the most important source of energy for many organisms. Like we also need carbohydrates. Every day we eat starch and to get the energy. Plants, however, you use the sun's energy to convert carbon dioxide into carbohydrates. There is a difference. So, plants actually make the carbohydrates for us, and then we eat the plants and get the starch from it. And the many organisms, as I said, that cell wall, that is written in the last stage, that the cell wall also is important. The carbohydrates play a very dominant role. So, carbohydrates have many important roles in our life, okay, or in the living system. Store of energy, storehouse of energy, and it is a precursor of many biomolecules, and then even it is a precursor of the many fatty acids that we synthesize, like palmitic acid, stearic acid, those things, and it is a component of the cell wall, and it is also gives the energy to the plants also. Okay. Now, let us go to the lipids. As I said, lipids are the most variable group. I told you already, variable group of molecules, because the definition of lipid is not based on the structure. That is why we have a varied class. Lipids definition is something which is soluble in organic solvents and which is insoluble in water. So, there could be several different types of molecules. That is why we have different varieties of lipids. It could be fatty acids, it could be a steroid, it could be a terpene, and I will show you some molecules which are lipids. The molecules are shown here, like this is the first one is shown on the left, that is called a tristearine. Actually, that is known as triglyceride in general. Triglyceride is basically the triester of glycerol, and the ester, the acid part is a fatty acid. So, in this case it is stearic acid, because the acid comes from stearic acid. So, this is called tristearine. It is a fat, it is a soluble in organic solvent. Then you have on the right side the cholesterol. Cholesterol is a steroid, okay, and that is also a lipid, because you see there is no functionality in true sense, only there is a double bond and others are all carbons. So, in that way it is a very non-polar molecule, cholesterol. Then you have other different types of esters, like this something is cetyl palmitate, a wax kind of thing, and then you have a terpene, which is alpha pinene, is shown on the right side at the bottom left. Okay. So, these are some of the examples of lipids. Different types of saturated and unsaturated fatty acids are present, can be biosynthesized. The ones which dominate in the biological world is this lauric acid that starts with C12, that means 12 carbon atoms are present there, then myristic acid, palmitic acid, stearic acid, and the C20 fatty acid is what is called arachidonic acid, or actually arachidic acid in this sense, that is the name, but the derivative that comes from this is called arachidonic acid. On the bottom there is a block, this a 5 carbon unit we can see, there are 5 carbons with a diphosphate, and this is called isopentenyl diphosphate. This is one of the precursor through which we make the steroids, we make the terpenes. Okay. And this also has a very interesting biochemical way of making. Okay. Remember what I said, when the synthesis takes place in biology, that is what is called biosynthesis. Okay. And when it takes place in the laboratory, in our test tube, we call it synthesis. Organic synthesis is just plain synthesis that we call, and in biology whatever molecules we synthesize, that is called an anabolic process. Anabolic process means we make molecules, and the reverse is also there, that is called catabolic process or catabolism, where we break down molecules into smaller fragments. Okay. Both processes are essential in our body. This is a steroid structure shown here, and you see the water steroids. By definition, the steroids are basically having these 3 6-membered rings A, B, C, and that is fused with a 5-membered ring, which is shown as D. So, this is what is called, first of all, this is a derivative of phenanthrene, but phenanthrene no double bond, without any double bond. So, this is called parhydrophenanthrene. When you have angular fusion between 3 rings, 3 6-membered rings, that is called phenanthrene. If it is linear, that is called anthracene. So, steroid is actually called cyclopentano parhydrophenanthrene, that is what is a steroid. Okay. And this steroid, as I said, I showed in the previous slide that it was the isopentenyl pyrophosphate or diphosphate, that is the precursor of a steroid. Okay. In molecular modeling, there is a structure shown here, a model of this steroid, and it sees that you have so many atoms crowding together. Okay. And there is, in this molecule there is no functionalized carbon. Okay. This is also another steroid molecule. This is called prostaglandins. The prostaglandins are basically, again I told you that there is a C20 carboxylic acid, which is called, if it is fully saturated, that is called arachidic acid, but if there are double bonds present in that, that is called arachidonic acid. And from this C20 arachidonic acid, prostaglandins are synthesized in the body. So, that is biosynthesized in the body, and there are, when they are biosynthesized, you see if you count the carbons, it will be, there will be 20 carbon atoms, including the carboxylic acid, and you have different varieties of prostaglandins, but the definition is that it will have a 5-membered ring as is shown on the left side and along with 2 side chains. One side chain you have the carboxy terminus. This is called prostaglandin E1, and depending on the different geometries and the position of the double bond, you have different prostaglandins. They are extremely important. They have extraordinary range of physiological effects, including an ability to lower the blood pressure. So, they have a role in blood pressure, affect blood clotting, they are important. They are also, they regulate the kidney function, they control inflammation. So, when we have inflammation, we have pain, then what medicines we take, they are mostly like aspirin, they stop the biosynthesis of the prostaglandin. And another is that stimulate uterine contractions during childbirth. So, prostaglandin is also given for that. So, this is also very important, and in the 1970s when prostaglandins were discovered, it was thought that prostaglandins will solve many of the problems of many of the diseases that we have. Yes, it has solved many of these problems. There are anti-inflammatory agents, there are agents which help childbirth, there are agents which help in lowering of blood pressure. So, definitely they are extremely important, and but they cannot solve all the problems which was at that time overhyped in the 70s. Okay. After this we come to the nucleic acids. Now, this is I tell you that these are very general topics. We are saying about these molecules of life. Nucleic acids are they composed of basically they are polymers of nucleotides. Now, nucleotides are what? Nucleotides are basically having a phosphate, having a sugar and having a base. So, base, sugar and a phosphate, and they are linked one after another, and that make a strand, what is strand of nucleic acids. And there are different types of nucleic acids. Broadly there are 2 types, deoxynucleic ribonucleic acid and then the ribonucleic acid. Okay. And if I say what is the genetic material of the normal eukaryotic cells like the higher organisms, then DNA has to be. There is no eukaryotic organism where DNA is not the genetic material. DNA is the genetic material in all eukaryotic cells. This eukaryotic cells means a higher level of organism, because the organism it starts with unicellular systems, and where there was basically only a cell, only a membrane, and all cells are basically same in that case. Then the higher organisms started growing. Okay. So, we have a distinct class, prokaryotes and eukaryotes. Prokaryotes like the bacteria, they have different they do not have actually compartmentalized cell cytosol. Cytosol means what is present in the cell, but in eukaryotes we have compartmentalized systems, and there is a nucleus in a cell, and in the nucleus this deoxyribonucleic acid is present. Again I repeat, this is made up of the base, sugar, phosphate, that is the building block called the nucleotide or deoxynucleotide, to be precise if it is DNA, and the bases are of 4 different types, and then finally and the RNA also there are 4 different bases, only there is a difference between DNA and RNA, that RNA has a base which is called uracil. In DNA that uracil is replaced by what is called thymine. But this is just a very general slide that I am showing, and in the bottom the structure, the 3-dimensional structure, the very famous structure which was proposed by Watson and Crick, and that is called the double helical structure of a DNA. Okay. Then comes the other small molecules that we have talked about, because the biological domain is basically 2. One is the large molecules, another is the small molecules. The small molecules are also equally important. Okay. Equally important means like neurotransmitters, which gives the signals that what to do, when to do and when to stop. All these things are controlled by small molecules or sometimes large molecule interactions are also possible, but the whole process is called signal transduction. This signal transduction is controlled by the central nervous system, where we generate the neurotransmitters, which are basically very small molecules. Even amino acids are also neurotransmitters. I will show you some of the structure of neurotransmitters. So, you see acetylcholine, it is a so simple structure, it is very small molecule, but extremely essential. If we have acetylcholine which is not secreted properly, which is the level not maintained properly, then we suffer from many diseases. In fact, there are drugs, there are molecules which are acetylcholine, which controls or lowers the acetylcholine to such a level that the organism dies. Okay. Then we have dopamine, that is also a very simple molecule. So, these are very simple, the simplest of the molecules that are again essential for the functioning of the biological system. Remember, the biological system is made of macromolecules, which are called molecules of life. Okay. 4 classes I have shown, but then once you have the structure, you know that it is like a house, that you build the house, but then the house has to be operational. You need electricity, you need water, you need so many things, and these small molecules are basically functioning like this, what is electricity, what is light, because without that the whole house will be the persons who are living in the house, their life will be miserable. So, also these molecules are like that. If we cannot maintain, if we cannot have these molecules in our body, then these our life will also become miserable, and at some point of time there may be serious complications where people die out of, they lose their memory, all these things can happen. Okay. So, these are there are other small molecules, different types of small molecules showing there are these vitamins. We talk about vitamins, these are also extremely essential for molecules, for making molecules, for catalyzing reactions. They help many enzymes to carry out the reactions that they are supposed to do. So, I think that basically these are some of the molecules which are medium sized, means we have a small molecule, we have large molecules. In between, in the interface, some molecules are there, which are for molecular weight is, you cannot classify them as very macromolecules, you cannot classify them as very small molecules. Okay. So, they are medium sized molecules, and I have shown three important molecules here. Mostly they are, you see, they are made up of amino acids. Just I want to point out the one which is on the right side, that is called bradykinin. This is a hormone which again controls the blood pressure. It is a vasodilator, that means it dilates the blood vessel, so that the blood flow can become smooth, and that causes the blood pressure to drop. Okay. So, that is, now we have gone through all these different molecules that are involved in the biological world. This is a very minuscule number of molecules I have shown to you. There are thousands and thousands of molecules that are present in the biological system, but this is just now the introductory, the molecules whose chemistry we are going to describe in the subsequent lectures. Okay. Thank you.